Welcome, everyone. I'm excited to kick off this presentation on the fascinating topic of the cell cycle and cell division. Today, we'll explore how cells grow, divide, and ensure the proper functioning of living organisms. We'll cover the main stages of the cell cycle, the process of cell division, and why it's so essential for life. So, let's dive in and discover the amazing world of cells together. Let's take a look at the chapter outline for today's lesson. This slide provides a roadmap of what we'll be covering. First, we'll start with the basics of cell division and the reasons why cells need to divide, which is fundamental to understanding growth and reproduction. Then, we'll explore the detailed phases of the cell cycle, including interphase, the mitotic phase, and the specific stages within mitosis such as prophase, metaphase, anaphase, and telophase. We'll also discuss cytokinesis, the final step where the cell actually splits into two. Throughout the presentation, we'll connect these concepts to the broader theme of how cells control their division, ensuring everything occurs properly. We'll look at both eukaryotic and prokaryotic cell division to see the differences and similarities. Plus, there will be self-quiz sections to test your understanding along the way. And of course, we'll refer to key references that deepen our knowledge. By the end, you'll have a comprehensive overview of the cell cycle, its regulation, and why it's crucial for life. This outline serves as your guide through the complex but fascinating process of cell division. Let's talk about the regulation of the cell cycle, which is a crucial part of understanding how cells divide and function properly. The cell cycle isn't just a random process. It's tightly controlled by various checkpoints and signals to ensure everything happens at the right time. These control mechanisms prevent errors like uncontrolled growth or mutations, which can lead to diseases such as cancer. By understanding how the cell cycle is regulated, we can better grasp how cells maintain healthy growth and division, and how disruptions in this regulation can cause problems. This topic is fundamental because it connects directly to the overall theme of cell division and the importance of proper cellular function in living organisms. Let's talk about the cell cycle. The cell cycle is the series of events that take place in a cell as it grows and divides. It's essential for growth, repair, and reproduction of cells. Understanding the purpose of cell division helps us see why this process is so vital. It allows organisms to develop, maintain healthy tissues, and reproduce. Now, cell division differs between prokaryotic and eukaryotic cells. In prokaryotic cells, which are simpler, division is usually through a process called binary fission, where the cell splits into two identical cells. In eukaryotic cells, the process is more complex, involving multiple stages that make up the cell cycle. Let's define cell division as the process where a parent cell divides to produce two or more daughter cells. The cell cycle, on the other hand, is the entire series of events that a cell goes through from one division to the next. By the end of this session, you'll be able to explain these key points thoroughly, connect them to the larger picture of biology, and understand how cell division supports life at every level. Now, let's look at the cells after cell division. These are called daughter cells, and they are essentially the new cells that result from the process of division. It's important to understand that cell division is a fundamental process for life, allowing organisms to grow, repair damaged tissues, and reproduce. In eukaryotic cells, which include plants, animals, and fungi, the process is a bit more complex than in prokaryotes. Before dividing, the cell needs to replicate all of its DNA across multiple chromosomes, ensuring each daughter cell receives a complete set. Additionally, the cell's organelles are duplicated so that each new cell can function properly. Eukaryotic cell division includes processes such as mitosis, which is primarily for growth and repair, and meiosis, which is essential for forming sex cells or gametes. These processes are crucial because they contribute to genetic diversity and reproduction. On the other hand, most prokaryotic cells, like bacteria, divide through a simpler process called binary fission. In binary fission, the cell essentially copies its DNA and then splits into two identical daughter cells. It's a straightforward process, but it accomplishes the same goal: creating new cells from pre-existing ones. Remember, regardless of whether the cell is prokaryotic or eukaryotic, all cells originate from existing cells through division. This continuity is fundamental to the life cycle and growth of all living organisms. If cells grow without limit, an information crisis would develop because DNA cannot meet the growing needs of the cell. As the cell gets larger, it needs to produce more proteins and other molecules, but the DNA's capacity to store and process that information doesn't increase proportionally. This creates a bottleneck, or what we call an information crisis. This is why controlling DNA overload is so important. The cell must find ways to manage this, and one key aspect is the exchange of materials across the cell membrane. Food and oxygen need to cross quickly to support the cell's functions, while waste products must be expelled efficiently. If the cell becomes too large, these exchanges happen too slowly, which can lead to cell death, so size really matters. Cells divide for several crucial reasons: growth, development, repairing damaged tissues, and reproduction. By dividing, cells can maintain an optimal size, ensuring that the exchange of materials remains efficient. This process also helps prevent the cell from becoming too large and overwhelmed by its own DNA. So, in essence, cell division is a vital process that keeps cells functioning properly, supports the overall health of the organism, and ensures that the exchange of materials occurs smoothly, preventing the risks associated with uncontrolled growth. Now, let's look at what happens during prokaryotic cell division. First, each new daughter cell is genetically identical to the parent cell, which means the genetic information is faithfully copied and passed on. This is crucial for maintaining consistency within bacterial populations. Next, we have cytokinesis, or separation, where the cytoplasm divides, resulting in two distinct cells. This process ensures each new cell has the necessary cellular components to function properly. Then there's chromosome segregation, where the chromosomes are separated and move toward opposite ends, or poles, of the cell. This movement is essential because it ensures each daughter cell inherits a complete set of genetic material. The DNA replication step is also vital. It involves copying the DNA so that each chromosome consists of two identical sister chromatids before division begins. All these processes are part of what we call binary fission, which is the primary method of reproduction in prokaryotes. To sum up, these major steps, DNA replication, chromosome segregation, and cytokinesis, work together seamlessly to produce two genetically identical daughter cells, helping bacteria grow and thrive. Now let's talk about what happens after a cell divides. Each daughter cell is genetically identical to the parent cell, meaning they have the same DNA and genetic information. This is crucial for growth, repair, and maintaining the stability of an organism. Next, we have cytokinesis, which is the process where the cytoplasm divides, resulting in two new daughter cells. Think of it as the final step where the cell physically splits into two. Then there's mitosis, which involves four main stages: prophase, metaphase, anaphase, and telophase. These stages ensure that the chromosomes are accurately duplicated and evenly distributed between the two new cells. Mitosis is a type of cell division that produces two daughter cells, and importantly, each of these cells has the same number and kind of chromosomes as the original parent cell. This process is a key component of eukaryotic cell division, allowing organisms to grow, develop, and heal. It's a highly regulated and precise process that maintains genetic consistency across cells. Now, let's talk about how each daughter cell is not exactly the same as the parent cell. This is a key point because it highlights the importance of genetic variation and how it plays a role in development and evolution. When cytokinesis occurs, the cytoplasm divides, which results in two new daughter cells. Think of cytokinesis as the actual division of the cell's contents, ensuring each new cell gets the necessary components. Moving on to mitosis, this process involves four main steps: prophase, metaphase, anaphase, and telophase. These steps work together to ensure the accurate distribution of duplicated chromosomes into each daughter cell. It's a tightly regulated process that maintains genetic consistency during cell division. Now, it's also important to distinguish between mitosis and meiosis. Mitosis results in two identical daughter cells, mainly for growth and tissue repair. In contrast, meiosis is a special type of cell division that produces four genetically diverse daughter cells, which are essential for sexual reproduction. Understanding these processes helps us appreciate how cells multiply and diversify, supporting the overall theme of eukaryotic cell division and its vital role in life. Now let's take a closer look at the mitotic phase, starting with interphase. This is a crucial period in the cell cycle where the cell is preparing for division. The cell cycle itself has two main parts, one where the cell is actively dividing and another where it's preparing for that division. After a cell divides, it begins a new cycle, starting all over again. During interphase, the cell is busy with important activities. It's growing, developing, and most notably, replicating its DNA so that each new cell will have a complete set of genetic material. You can think of this as a period of arise and divide. The cell first arises from a previous division, then goes through stages of growth and preparation, culminating in DNA replication. This continuous sequence of events, from when a cell first appears after division to when it eventually divides again, is what we call the cell cycle. Understanding this cycle is fundamental because it explains how organisms grow, repair tissues, and reproduce cells. So, in summary, interphase is the phase where the cell is building up everything it needs to successfully divide, highlighting its role as the preparatory stage before the actual division occurs. Now, let's move on to the G2 phase, which is a crucial part of the cell cycle. During this stage, the cell continues to grow and prepare for division. All the structures needed for mitosis, like the spindle fibers, start to form. At the same time, the cytoplasm is expanding, providing the necessary components for the upcoming cell division. Organelles are doubling in number, ensuring that each new cell will have enough of these vital components. One of the most important events in G2 is the production of two sister chromatids for each chromosome. This means that the DNA has been replicated, creating identical copies that will be separated into the two new cells. This replication occurs during the S phase, which is part of interphase, the period when the cell is not actively dividing, but getting ready. It's important to remember that interphase takes up about 90% of the cell cycle, making it the longest phase. It's a period of growth, DNA replication, and preparation that sets the stage for successful cell division. So, in essence, G2 is all about finishing up preparations, building the structures, doubling organelles, and ensuring the DNA is fully replicated, so the cell is ready to move into mitosis and divide properly. Now, let's take a closer look at the cell cycle, which is fundamental to understanding how cells grow and divide. The cell cycle is a series of well-organized phases that a cell goes through to divide and produce new cells. It all begins with the interphase, where the cell prepares for division by growing and copying its DNA. During this phase, the cell spends most of its time making sure everything is ready for the next steps. Once the cell has duplicated its DNA, it enters mitosis, which is the process where the duplicated chromosomes are separated into two new nuclei. This is a critical step because it ensures each new cell will have the correct genetic information. After mitosis, the cell enters cytokinesis, where the cell physically divides into two daughter cells. Understanding the cell cycle is crucial because it connects to many broader themes in biology, like growth, development, and even how cancer occurs when this cycle goes awry. Recognizing these phases helps us see how life perpetuates at the cellular level, maintaining the health and function of entire organisms. Now let's take a closer look at the mitotic phase, or M phase. This is the stage where the cell actually divides. When we talk about mitosis here, we're specifically referring to the division of the nucleus. It's a critical process because it ensures that each daughter cell receives an identical copy of the genetic material. During this phase, two main things happen. First, the division of the nucleus, which is what we call mitosis. And second, the separation of the cytoplasm and the newly formed nuclei into two separate daughter cells, a process known as cytokinesis. So, in simple terms, mitosis involves splitting one nucleus into two genetically identical nuclei, ensuring the genetic information is preserved. The mitotic phase is the stage when this cell division really kicks into gear, allowing the cell to produce two new, identical cells. Connecting this to the bigger picture, the mitotic phase is essential for growth, tissue repair, and overall cellular reproduction within the organism. Without this precise division process, proper development and maintenance of tissues wouldn't be possible. All right, let's take a closer look at the stages of mitosis, starting with the order: prophase, metaphase, anaphase, and telophase. These four stages are collectively known as the PMAT stages, and they make up the first mitotic phase. During prophase, the chromosomes begin to condense and become visible, and the nuclear envelope starts to break down. It's like the cell is preparing itself for division. Next, in metaphase, the chromosomes line up at the cell's equator, also called the metaphase plate. This alignment is crucial because it ensures that each new cell will get an equal and accurate set of chromosomes. Then, in anaphase, the sister chromatids are pulled apart by the spindle fibers and move toward opposite poles of the cell. This separation is vital for genetic consistency between the daughter cells. Finally, during telophase, the chromosomes reach the poles, and new nuclear envelopes form around each set. This marks the near completion of nuclear division. These stages are divided into two main mitotic phases, which work together to ensure that the cell divides correctly and evenly. Understanding these steps helps us grasp how cells replicate accurately, maintaining the integrity of genetic information through cell division. Now, let's take a closer look at what happens during interphase, which is the preparatory phase before cell division. First, you'll notice the term centrioles here. These are small structures located near the nucleus that play a crucial role in organizing the spindle fibers during cell division. They help ensure that chromosomes are evenly divided between the two new cells. Next, we see the nuclear membrane. During interphase, this membrane is intact, enclosing the nucleus and protecting the genetic material inside. It's like a security barrier that maintains a controlled environment for the DNA. Moving on to the nucleolus, which appears as a dense region within the nucleus. The nucleolus is primarily involved in producing ribosomes, essential for protein synthesis. Its activity is especially high during interphase as the cell prepares for division. And finally, the chromatids. You'll notice there are two of them here. These are the duplicated forms of chromosomes, and during interphase, the DNA replication occurs, resulting in these sister chromatids. They stay connected at a point called the centromere until they're ready to separate during mitosis. So, in essence, interphase is all about the cell preparing itself, growing, replicating its DNA, and getting ready for the division process ahead. This phase is fundamental because it sets the stage for accurate and efficient cell division, ensuring each new cell gets the right genetic information. Now, as we move further into early prophase, you'll notice that the chromosomes start to coil and thicken, making them more distinct from one another. This is an important step because it helps us see the chromosomes clearly under the microscope. Each chromosome now appears as two identical halves called chromatids, which are connected at a central point known as the centromere. This connection is crucial because it ensures the chromatids stay attached until they are separated later in cell division. At the same time, a structure called the spindle begins to form. This spindle is made of microtubules and will play a key role in pulling the chromosomes apart later on. Also, you'll see the centrioles. These are the small cylindrical structures, separating and starting to move toward opposite ends of the cell. This movement helps organize the spindle fibers and sets the stage for chromosome movement. Additionally, the nucleolus, which is involved in ribosome production, starts to disappear. This is a normal part of prophase, signaling that the cell is progressing through the early stages of division. Overall, These changes are vital for preparing the chromosomes for separation and ensuring the cell division process proceeds smoothly. Now, at this stage, the chromatids are really starting to get organized because they are attached to the fibers of the spindle. This attachment is crucial for ensuring that each new cell will receive the correct set of genetic information. By this point, the spindle itself is fully formed, which means the microtubules have arranged themselves properly to facilitate chromosome movement. You'll also notice that the centrioles have moved to opposite poles of the cell. This positioning is essential because it helps guide the spindle fibers and ensures the chromosomes are pulled apart accurately. Speaking of microtubules, they've now formed a complete spindle, acting like a sort of scaffolding that connects the chromatids to the centrioles and helps facilitate their movement. Additionally, the nuclear membrane has broken down or fragmented, which allows the microtubules to invade the nuclear area. This invasion is vital because it clears the way for the chromosomes to be aligned and separated efficiently as the cell prepares for division. Overall, these steps are key in late prophase, setting the stage for the next phase where the chromosomes will be aligned and separated into two new cells. Now, let's take a closer look at what happens during metaphase. The spindle fibers play a crucial role here. They will push and pull the chromosomes, helping to align them properly. Each chromosome is connected to a spindle fiber at its centromere, which acts like a central hub holding the sister chromatids together. You'll notice that the centrioles move toward the polar ends of the cell and extend spindle fibers that reach out to connect with the chromosomes. This setup ensures that the chromosomes are lined up at the metaphase plate, the imaginary central plane of the cell. This precise alignment is vital for the next step of cell division, ensuring that each new cell will receive an identical set of chromosomes. So, in essence, metaphase is all about organization and preparation for the equal separation of genetic material. Now, during anaphase, the microtubules start to shorten, and this action pulls the chromatids apart, moving them toward opposite sides of the cell. This process ensures that each new cell will get an identical set of chromosomes. By the time anaphase ends, both ends of the cell have completed sets of chromosomes that are equal in number and complete. At the same time, partial division of the cytoplasm begins, which is an important step toward final cell separation. The separated chromatids, now considered individual chromosomes, move to their respective poles. This movement is crucial for maintaining genetic consistency. Remember, the paired chromosomes, or sister chromatids, are finally separating in this phase. This separation guarantees that each daughter cell will have the correct genetic information. Overall, anaphase is a key moment in ensuring that the genetic material is accurately distributed, setting the stage for the formation of two healthy, complete daughter cells. Now, as we reach the final stages of telophase, the nuclear membrane starts to form again around each set of chromosomes, creating two separate nuclei. At the same time, the nucleolus reappears inside these new nuclei, which is a sign that the cell is returning to its usual functioning state. You'll also notice that the cell itself begins to pinch inwards, a process called cytokinesis, which ultimately leads to the formation of two distinct, identical daughter cells. During this process, spindle fibers, which helped separate the chromosomes earlier, disappear, indicating that their job is done. The nuclear envelopes are re-establishing, enclosing the genetic material. The chromosomes, which were tightly coiled to facilitate movement, now uncoil back into chromatin, making it easier for the cell to carry out its normal functions. Finally, the chromosomes are positioned at opposite poles of the cell, ready for the cell to complete division. This entire process in telophase is crucial because it ensures that each new cell receives a complete set of genetic information, maintaining the integrity of the organism's genetic code. It's the final step before the cell fully splits into two, closing the cycle of cell division. Now, let's take a closer look at how cytokinesis differs between plant and animal cells. In plant cells, after the chromosomes have separated during mitosis, a cell plate begins to form in the middle of the cell. This cell plate acts as a new dividing wall, ultimately separating the two daughter cells. It's important to note that this process usually occurs at the same time as telophase, although during telophase itself, the division isn't yet visible. The initial formation of the cell plate actually starts during telophase, setting the stage for complete separation. In contrast, animal cells divide differently. Here, we see a cleavage furrow forming. Think of it as a contracting belt pinching the cell in the middle. This furrow deepens until the cell is split into two separate daughter cells. This process typically happens right at the end of mitosis, ensuring that each new cell has its own complete set of genetic material. A key point to remember is that, regardless of the method, the daughter cells are genetically identical to each other and to the original parent cell. This is crucial for maintaining the integrity of genetic information across cell generations. So, in summary, plant cells form a cell plate to divide, while animal cells use a cleavage furrow to accomplish the same goal. Both processes are essential for proper cell division and tissue growth. Now, let's talk about mitosis, which is a crucial part of cell division. Mitosis is the process through which a single cell divides to produce two identical daughter cells. This process ensures that each new cell has the same genetic material as the original, maintaining consistency across our tissues. It's important because it underpins growth, tissue repair, and overall cellular maintenance in our bodies. During mitosis, the DNA is carefully duplicated and evenly distributed, which is vital for healthy cell function. By understanding mitosis, we see how our bodies keep everything functioning smoothly and how cells replicate accurately. This process ties directly into the larger theme of how living organisms grow and heal. It's truly fundamental to life. All right, so now we've reached the part where we actually design an education game from here. As you can see, the slide lists the phases starting at the top, with D, B, C, and then E and A at the bottom. These phases represent the key steps we need to consider when creating an engaging and effective educational game. The first step is to identify the core learning objectives. What do we want the learners to achieve? Then, we move to designing the game mechanics and narrative to support those objectives. It's crucial to keep the game engaging, but also aligned with the educational goals. Next, we think about how to incorporate feedback and adaptive challenges, making sure the game responds to the learner's progress. This ensures that the experience remains motivating and customized to individual needs. Finally, testing and refining the game based on actual user feedback is essential before full deployment. Remember, these phases are interconnected and iterative. They build on each other to create a cohesive learning experience. By following these steps, we can develop a game that's not only fun, but also educationally impactful. Let's take a closer look at some key structures involved in cell division. First, we have the centromere, which is the part of the chromosome that holds sister chromatids together. It's crucial during cell division because it's where the spindle fibers attach. Speaking of spindle fibers, these are the structures that help pull chromosomes apart during mitosis. Next, we see the centrioles, which are involved in organizing the spindle fibers. Now, as we go through this diagram, you'll notice labels like Z and Y. These are specific points or structures we need to identify. Similarly, there are points labeled W and X that are important for understanding the process. The sister chromatids are identical copies of a chromosome that are connected at the centromere. During cell division, these sister chromatids are separated to ensure each new cell gets a complete set of genetic information. Now, the key phases of cell division are also referenced here. These include prophase, metaphase, anaphase, and telophase. Each phase has distinct characteristics, and understanding these helps us see how chromosomes are accurately segregated. Finally, this is a self-quiz slide, so I encourage you to think about how these structures work together during each phase to facilitate proper cell division. Recognizing these components and phases is essential for understanding how life propagates at the cellular level. Now, let's talk about the final checkpoint, which is the mitotic spindle checkpoint. This checkpoint ensures that all chromosomes are properly aligned at the mitotic plate before the cell proceeds with division. It's a critical control point because if chromosomes aren't correctly lined up, it could lead to errors in chromosome segregation, which can cause serious problems like genetic mutations or cell malfunction. Moving back to the earlier points, we have the G1 checkpoint. This is where the cell decides whether it's ready to divide or not. If conditions aren't right, the cell can halt its cycle here. Then, there's the S checkpoint, which makes sure that the DNA has been properly replicated before moving forward. Proper DNA replication is essential for maintaining genetic stability. And finally, the overall control of the cell cycle is tightly regulated by proteins called cyclins. These cyclins act like molecular switches, turning certain processes on or off at the right times during the cycle. They help ensure that each phase happens in the correct sequence and that the cell only divides when everything is properly prepared. All of these checkpoints and regulatory proteins work together to keep cell division accurate and controlled, which is vital for healthy growth and development. Let's take a moment to appreciate the intricacies of prokaryotic cell division. As you can see here, this process is fundamental for the growth and reproduction of bacteria and other single-celled organisms. Unlike eukaryotic cells, prokaryotes typically divide through a process called binary fission, which is simpler but highly efficient. It involves the duplication of the DNA, then the cells splitting into two genetically identical daughter cells. This process highlights how life can be sustained and propagated even in the simplest organisms. It's also a great example of how cellular mechanisms are adapted to different organismal complexities. Understanding prokaryotic cell division not only deepens our knowledge of microbiology, but also connects to broader themes in biology, such as growth, reproduction, and evolution. And as always, I want to give credit to the creators of this informative image, Mariana Ruiz Villarreal and the CK12 Foundation, whose work helps us visualize and understand these fundamental processes.